Greetings everyone, my name is Joe Francis and I've taught at the college level for a number of years in the areas of microbiology and public health and immunology. The areas I really love are microbiology and immunology down to that cell level. And what we certainly see is God's handiwork. We see His blessings in a way, if you think of the immune system, keeping us going and keeping us healthy. Microbes, though, are a bit of an enigma because they look designed. They have features that show incredible design, yet they are so harmful to human life when we think about the diseases they cause, like strep throat and pneumonia. Both can be killers to many people every year. So I'm asking this question to start out here: why are there bad microbes in God's perfect creation? And that's kind of a leading question because we know God's perfect creation has been marred by sin. And so how do we think about microbes then? How were they changed? Were they changed? Did they occur after the fall? How do we address this question? So let's take a look at the premise of this presentation, and that is, I believe there's good evidence biblically and scientifically to support the idea that all existing microbes are connected to the beneficial microbes that were formed during the creation week. Again, I'm taking that position because because microbes appear to be designed, and we see God's design in His creation and also see that He called it to be very good. So then we can expand this premise to say maybe some of the bad microbes could be altered good microbes. Maybe it didn't take very much to alter them, to have some of their secretions that maybe help us digest food than to cause disease, let's say. Or some of them were not changed but found themselves to be in a place they were not meant to be. In creationism, this is often called the displacement theory. And it's a general theory where we'd hope to flesh it out a little more. We could think of weeds in your garden. Some of the weeds in your garden might be plants that others are trying to grow for a certain reason, but because they're in your garden, we call them weeds, we call them disruptive, and they can be. So how do we think about these things? Are some things that God created in the wrong place? With microbes, though, however, we have to deal with illness. We have to deal with disease. And we just went through the COVID pandemic, and thankful that so many lives were probably spared by medical help and that sort of thing. But the death toll is around 7 million for COVID. However, if we go back to 1918 and the Spanish flu, we find that 50 million people worldwide were killed. That is the death rate, the death number. And it's striking that many people were removed from the earth. Their lives are removed from the earth. So pandemics have definitely changed the world, the way we look at it, the way we think about microbes. And there's a book written by Jonathan Kennedy on this very topic called Pathogenesis, A History of the World in 8 Plagues, an interesting discussion, again, showing just how devastating they are. So again, why are they here? How did they change? So to begin our discussion, to look into this idea about where bad microbes came from, we're going to take a look at some of the classifications of microbes first and find out how bacteria fit into this group. And we can look at least 5 microbe types, 5 basic microbe types, but there's probably more. And we can look at least at 5 basic types that you'll find in the microbiology textbooks. I asked AI to draw them. And so what it came up with are some drawings that are very close to what we find in microbiology textbooks and how they're classified. We have 4 types on this slide, and the 5th one will be on the next. So let's look at these 4 types. You can see the big spheres right here, bacteria. And I would say we know they're bacteria compared to viruses because bacteria are bigger than viruses. So AI did not do a bad job in making a smaller sphere, and viruses can take this spherical shape. So, and the big bacteria, the round bacteria, also called cocci, have these fimbriae or arms or tentacles coming out. Technically they're called fimbriae, sometimes they're called flagellum, pili, they're even called pili, there's different types. that is real. They have coatings of those on their surface in a way to sense the environment, for even twitching some of them to move around. Viruses, again, can be round coccozoid shape, but what they're not showing here, what AI didn't show, is that some viruses can take a very geometrical shape, a very rigid, like almost looking like little spaceships. That's the way I think of them. So viruses can take on many shapes, but technically they are smaller than bacteria in general. We have a rod-shaped bacteria right here. This is a famous shape because the famous E. coli that we talk about in medicine and in health takes that shape, and our gut has a lot of E. coli. And so then we can look at algae and fungus, and what's interesting about both of those is they can be shown as single cells. Maybe this red one right here is a single-celled fungus, and a green one is a single-celled algae. But they also can take on a plant-like structure. They can join together some of those individual fungi and algae and make plant-like structures, which is really interesting. And sometimes they can do this at will, depending on the stimulus in the ocean or wherever they're at. And so that's intriguing, kind of like shapeshifters in a way. And so AI did not do a bad job. I don't know what this creature is right here. It looks like it's holding eggs with smaller versions of creatures inside of it. It's very possible that that shape and creature exists as a microbe. So this is interesting. So the fifth classifications are protozoan structures, protozoan creatures, and they're single-celled, but they have a lot more complexity inside of them, what we're going to talk about. And in fact, this rotifer can, and sometimes these protists are often referred to as pond organisms. We find them swimming in ponds. And so this rotifer is often kind of put in pictures of microbes when it's actually an animal. But some people kind of consider it a pond organism microbe. So just like biology in general, sometimes it's hard to figure out one creature from another in God's amazing creation. What I want to look at is this protist, which is called a ciliate. It's a ciliate because it has these little cilia fibers on it, again, probably for locomotion. But one of the interesting things about this ciliate is it's photosynthetic. It has green pieces. But this is interesting. Those green pieces are a bacterium called cyanobacteria. So here is a creature, a protozoan, that has engulfed a bacteria and used it to derive energy from its photosynthesis. What's even more interesting is this ciliate used its oral cavity or mouth to uptake those bacteria. But in reality, also it uses that mouth to take up bacteria to eat them. So for some reason, it discriminates the photosynthetic bacteria and doesn't destroy them in its gut, whereas it destroys the other bacteria. So what we're finding is this gets really mixed up. We're finding we can't just, the classifications are involving creatures that are mixing together, so they're called mixotroph. We're finding mixotrophs are probably more the norm than not, where two or more microbes are living together and in symbiosis. Symbiosis means they work together to survive. And so I just wanted to show you a little bit about classification because it's not necessarily straightforward, but bacteria have been probably the most studied of all the microbes. So we want to look at bacterial classification, design, and beneficial activities of bacteria. Again, addressing this question, where did bad microbes come from? And so let's take a look at bacterial classification. And they are classified as prokaryotes. Now this is an interesting word because it's driven by evolution theory. Evolution theory says, well, microbes are a lot simpler than human body cells or animal body cells. And so we say they say before. Prokaryotes or bacteria are before what? Prokaryote. Karyote describes the nucleus. The nucleus is where the DNA is inside the cell. It's membrane bound. Bacteria don't have a membrane bound nucleus. At least we haven't discovered one yet. So what they're saying is these are ancient creatures that evolved before the eukaryotes, which are the cells that have the nucleus within. And so this is interesting. So their naming has been driven by evolution. If we look at the prokaryotic cell versus the eukaryotic cell, we'll see the nucleus, the membrane-bound nucleus, the nucleus within. And in the prokaryotic cell, the before nucleus creature, the bacterium, we see DNA, which seems to be free as a circle, is circular. But what they're not showing here is the tremendous complexity of both of these structures. The eukaryotic cell nucleus is very well designed. It's an incredible machine because it has pores in which some nucleic acids are DNA and RNA. DNA is copied into RNA and RNA is let out. And you have this amazing discriminating pore that allows RNA to leave, and the RNA goes out, and that's what proteins are made from. We don't have that same structure in the prokaryotic DNA. There's no nuclear membrane, but it is so complex itself. It's called the nucleoid. They came up with a name for it. And it kind of shows an admission. This is ending up to be more complex than we thought. If we look at an electron microscope image of this, we find the DNA in the prokaryotic cell just filling that entire bacterium. And what we find are pathways, designed pathways, to make proteins in that DNA area and push them toward where they need to go. We're finding more complexity the more we study the prokaryotic cell. So here are some shapes and some pictures of bacteria, and we find the pill-like shape, or what's called the rod. We can find them growing in filaments. This is a lot of bacteria that are attached together, rod-shaped bacteria. Here's more rod-shaped bacteria. Here are spiral-shaped bacteria, and here are really spiral bacteria right here called spirochetes that can actually bore into human flesh and cause disease. We can even find bacteria that are photosynthetic and grow in strands and filaments. So a lot of different shapes. What we find in nature is really interesting. The microbes in nature tend to be in these big clusters. When we look at them under the microscope in the lab, we see them one by one. But when we look at them in nature, they look more complex, growing in big groups. And then down here, we have a human white blood cell. Its nucleus is stained in red. And the little dots are probably a pneumococcal bacterium which can cause pneumonia. You can see how small these little red dots are compared to the white blood cell. We can't see the exterior of the white blood cell. We can just see its inside nucleus, but you can imagine a round shape right there. So let's look at prokaryotes, the prokaryotic classification a little closer. What we find now are two groups of bacteria, the archaebacteria and the eubacteria. What's going on here? And why are we using this term "eu" again? It's getting confusing because we have eukaryotic cells, which have the internal nucleus. And so it's not actually clear. But I think one reason they put "eu" in front of the common bacteria, most of the bacteria I just showed you in the last slide were the eubacteria. The archaebacteria are considered to be a bacteria evolved before the eubacteria. Archae means ancient. And where does this idea come from? Here's the question. Why were archaebacteria chosen as the first evolved bacteria? Well, here's the reason why. The reason why is they're found in extreme environments. They're found growing in acids. They're found growing in Yellowstone National Park in thermal vents, thermal pools where the temperature's too high for any other life form to exist. They're found in environments where there's no oxygen. They can breathe methane. And so now these archaebacteria, we have discovered, we call them extremophiles. But why did they give them the name archaebacteria as a first evolved cell before the eubacteria? It has to do with the early Earth. The evolutionary theories about the early Earth were that pieces of exploding stars and other pieces of maybe meteorites came together. These pieces came together in our solar system and smashed together and formed the Earth. Now, if you can imagine large bodies of things smashing together, what are you going to have? You're going to have heat. So I've asked AI to draw what the early world would look like during the evolution of the Earth. And it does show this burning, molten planet that's being formed. I'm not sure why it put another planet, Earth, right in the middle there, a little one, but that's what AI does sometimes. But this isn't a bad idea based on that evolutionary idea. So think about that for a minute. Can you see where this thinking about the archaebacteria comes from? Yes, you see that the archaebacteria are extremophiles, and they would be more likely to evolve on this hot Earth, or this Earth that's cooling off, and was probably considered to be burning for millions of years before it cooled off. But here's where it gets even more interesting. If we look at this idea that archaebacteria evolved before eubacteria, the common E. coli and so on, in the eubacteria class, what we find is that there's complexities that set them apart. So I'm showing you here a plasma membrane. Okay, this is a membrane found on the outside of all bacteria, all eukaryotic cells, all the cells in your body. It's made of a phospholipid. So a phospholipid has an orangish shape head and two what we call fatty acid tails. Those fatty acid tails have no charged atoms on them, so they don't like to interact with water. They would rather interact with themselves rather than water. The polar head group is polar, and anything that has charge, if something's polar, it has a charge. So the polar head group likes to interact with water, because when water dissociates, it has charges. So the polar head group likes to interact with water, the fatty acid tails like to interact with each other. And if we actually take some phospholipids and dry them out in a test tube, you can do this, I've done this experiment, and add salt water and shake it up, you will actually make something that looks like this, kind of like what you would find when you mix up certain kinds of salad dressings that have oil and water or vinegar in them. You would find these little, round structures that are basically lipids or fats. They're sticking together and they're avoiding the water. So that's why you have to shake up that salad dressing to mix the components. So you can actually, but the difference here is that those fatty acid tails, the fats, are solid in the middle of that. Where a cell doesn't have solid fat, it has a membrane. Imagine this thing coating a whole cell, this bilayer. It has water inside. So how do we get there? Actually, I've done this in the laboratory. You can make what is called a liposome. A liposome is an artificial membrane that we can make. So what you can do is take these fat globules, like right over here. Let's say we made them with phospholipids and they're solid. Okay? They all just wind up into one big solid mass. What you can do is actually shake them up even more and force them through pores, and they will make a bilayer. That's interesting. You say, wait a minute, is that what happened in evolution? That's what they think. The problem here is we're skipping over a lot of complexity. The complexity that we're skipping over is the fact that a true plasma membrane actually has proteins in it, and it has pores in it, and all those things have to be in the right place, or water builds up and explodes it. So where are we going with this? Well, we're going to the fact that both archaebacteria and eubacteria have fatty acid tails. Excuse me, but the archaebacteria has a fatty acid tails that are combined together. So there's an interesting phospholipid. The phosphate group with the multicolors here is on the ends, and the fatty acid tails are in the middle, but you don't have a bilayer, you have a monolayer. So all archaebacteria have this monolayer, kind of looks like the bilayer, but you remember these are not attached. So I did some work looking at multiple biochemistry papers on this topic. And what they said is, to make an archaebacteria membrane is very complex. To make a eubacteria membrane is very complex. It's a different set of proteins. And therefore, it makes it unlikely that one of these evolved from the other, because just to make this membrane is much different than how you make this membrane. And you can't get there unless you have the proteins that put those together. So in living cells, proteins do everything. The nucleic acids we talked about hold the information to make a protein. So we talked about the nucleus being very important. That holds the information to make a protein. Then proteins do everything else. And what we find is a very separate set of proteins making this bilayer compared to that one. And even I think some evolutionists have admitted this is a problem. How do these even relate together? And then the story gets even more intriguing. So if we look at 3 large groups of creatures on Earth, we can place all the animals and plants in a big kingdom we would call the Eukaryota. Okay, a big group called the Eukaryota. And what is different? They have nuclei. They have that complex nucleus. The archaeans and bacteria don't have that membrane-bound complex nucleus. But here's what they did. What they found out, they did a test and they found that the archaeans are closer to the eukaryotes. Wait a minute. The archaeans should have evolved first based on the idea of the Earth, the formation of the Earth that was so chaotic. How did they get this arrangement showing that the typical eubacteria evolved before the archaeans? They got there because they compared the DNA of these 3 groups. And the archaean DNA sequence is closer to eukaryotes. And DNA sequence tends to rule every discussion on relationship. So this is interesting. They had to throw out the whole idea that the archaeans evolved first. Okay, so this is interesting. But we have these 3 groups, and this is where bacteria live, kind of both in the eubacteria group and the archaean group. So here's a summary so far of where we've been. Bacteria are one of the kinds of microbes. Bacteria are divided into 2 large groups, the archaic groups and the eubacteria groups. In the evolution view, eubacteria evolved as the first cell before the archaebacteria, according to evolution. There is good evidence to show that archaebacteria and eubacteria are not related by evolution. So what about the relationship between bacteria and eukaryotic cells? Let's look a little closer at that. So let's look at bacteria and design in the context of some bacterial form would have to evolve before this eukaryotic cell. Okay, and let's look at the difference between eukaryotes and prokaryotes at the cell design level. So in this eukaryotic cell, again, I've asked AI to draw this, and it didn't do a bad job. I was fairly impressed with this. It has a nucleus, kind of a pinkish nucleus inside. That's that membrane-bound nuclear material, the DNA. It has folded membranes everywhere, and that's what we find in eukaryotes. Outside that nucleus, we find folded membranes everywhere. What's going on there? And then we have the plasma membrane on the outside in green. The folded membranes are basically form pathways where proteins are made and packaged. They're made and packaged. So what happens is the nucleus has the information to make a protein. The protein is made from that information in the DNA sequence. Okay? Proteins are made outside the nucleus. They are then packaged in lipid vesicles so that they can be moved to other areas where they can be modified. So this blue is sometimes called the endoplasmic reticulum, the ER. And proteins are moving through and being packaged in lipid vesicles. And sometimes they're sent, well, they're often sent to the Golgi. That's another folded membrane in orange here. And what happens there is the Golgi says, you need to go to the plasma membrane. We need a protein there. You're the right protein. We're going to put an address on you. So the Golgi is like the post office. It stamps, take this to the membrane. And it keeps going through these membranes, and the membranes are then coating the protein until they get to the surface of the cell or other places. Then we have another folded area right here called the mitochondria. And the mitochondria is folded membrane, and in those folded membranes, energy is made called ATP. And that is used to run this whole process. So this is really interesting. I have no idea what the sponge is doing on there. We don't find a sponge in cells, so we're going to ignore that. But a fairly good job on showing the complexity of the eukaryotic cell. Let's look at the prokaryotic cell generated by AI, and I was very surprised how good this is. Again, these fimbriae or pili on the outside, these fimbriae can be twitching. Some of them twitch like this and cause it to move like oars. And then we have flagellum on the ends, and that's a rotary motor. A rotary motor that actually sends it forward. The motor is very complex. Then I was intrigued by the center of this bacterium because it does have strands. Probably we could say that's DNA. The information that AI is getting is interpreting that as what I would say DNA. But what really intrigued me are these small circular vesicles. This is really amazing, really fascinating, because about 15 to 20 years ago, some groups said, you know, prokaryotes, bacteria, are not supposed to have these membranes inside. And I should have given you another description, one more term. Let's back up real quick here. All these structures that are lipid are called organelles. Kind of a crafty way to refer to the organs like we have in our body, and cells are called organelles. So this ER, which did its packaging, is called an organelle. This Golgi is called an organelle. This mitochondria is called an organelle. Bacteria just don't seem to have those, or so we thought. So within the last decade or so, scientists said, wait a minute, when we look a little closer at bacteria, what we find are packages. What is going on? They're called BMC. BMCs, bacterial microcompartments. And what we're finding is they segregate material inside these protein walls. Why didn't we see them? Probably because we had a commitment to think that the prokaryotes were simpler. And now, if you look at the far right diagram, you see when you make a protein-like membrane, it's not as bendable as a phospholipid membrane. Okay, it's very structured. The proteins are very stiff compared to the phospholipids. And inside that one right there is all the proteins to perform photosynthesis, because some bacteria are involved in photosynthesis. This is remarkable. This is incredible. One of my schools I went to, Michigan State, they had a team there that made this discovery of BMCs. Wow, we miss it. Did we miss it because we adopted this idea that they were simple cells? I think it's very possible. So all that to say is bacteria are complex in their own ways. So bacteria are complex and show design, and that's one of the things that I wanted to try to figure out. And because of that, it appears to me that they were created in the beginning for something. And we can say something good, because God said everything was good. So because God made everything for a purpose, Proverbs 16:4, we can predict that God made bacteria for a reason. And so in 2003, I wrote a paper for the International Conference on Creationism called The Organosubstrate of Life, a Creationist Perspective of Microbes and Viruses, starting with this idea that God made them and what are they about. We ended up changing the name organosubstrate to the biomatrix. So we present evidence that they're everywhere, forming a matrix, and in many cases, they're life-supporting. So we want to look at some examples of this. So here's 3 areas we're going to look at, the bullet points. Bacteria are designed for beneficial activities on Earth on a global scale, And we're calling this the biomatrix. Here's some things we're going to look at. Bacteria are abundant on Earth and in all ecosystems. What we mean by ecosystems is where living creatures live and how they interact. Bacteria recycle nutrients locally and on a global scale. The global scale is real. They work on a global scale to produce things like oxygen for us. Bacteria can act as symbionts for humans and many animals and plants. I believe there's relationships between all of these 3 too, showing that biomatrix is kind of connected across the globe. So if we look at some numbers, if we have, and we're going to use the term microbiome. So microbiome is just a fancy way to say all the bacteria that live on the human body. That's a human microbiome. Or we could say, oh, there's a forest microbiome, all the bacteria and other microbes that live there. And so if we look at a number, we can even calculate if every human has 35 trillion, on average, 35 trillion bacteria, which is, we believe that is the number because the average human has about 35 trillion cells, total cells, eukaryotic cells, then we believe there's at least one bacterium for each of our cells. So that gives each human roughly 35 trillion, and if you multiply that times the population, you get this number, 2.7 times 10 to the 23. There's a lot of bacteria on people. But they're also abundant in extreme places, even below the continents. So this is another AI image showing bacteria below the Earth. And here's some recent interesting data. Some 200 to 600 octillion microbes live beneath our continents. That suggests analysis of the data from sites it's from analysis of data from all over the world, and even more live below the seafloor. Together they weigh the equivalent of up to 200 million blue whales. Subterranean diversity rivals that of the surface, with most underground organisms yet to be discovered or characterized. This is interesting. You say, why? Why is God putting these creatures down where we cannot even live? Well, if you think about those verses in Genesis, it says he filled the earth. He wanted to fill the earth, especially the aquatic things, the water. He talks about swarming things. And we see it. We see it even in the great depths of the earth. In fact, here is an extremophile. This would be an archaean called 𝘎𝘕𝘯𝘕𝘴𝘪𝘴, living 1.5 miles deep in the ocean, on the ocean floor, northeast Pacific Ocean near Puget Sound. And this black smoker is a vent on the floor of the ocean where some of the tectonic activity is going with the plates, and there's great heat involved. And they spew out this hydrogen, sulfide and methane, and nothing you would think would really live in it. But we find bacteria living in that toxic environment. And in fact, so many of them are eaten by some creatures not a few feet away that actually have armor on to actually live there. Here we see God saying, I'm going to fill the earth with bacteria. I'm going to have them live in these extreme environments. And then I'm going to have them as food for creatures that live near the extreme environments. Again, I think it goes back to the truth that he was saying. I want to fill this earth with living things. What's interesting about 𝘎𝘕𝘯𝘕𝘴𝘪𝘴 is it can survive temps over 250 degrees Fahrenheit. So your oven, that's kind of a low heat for your oven, but you don't want to be inside it, right? That's pretty high. You can get burned quite easy. So, and it can also survive, this creature can also survive being in an autoclave. In fact, it would like to live there. An autoclave is a sterilizer that's used in healthcare, used in hospitals and clinics. It's kind of similar to your mom's pressure cooker that you might have on the stove. But if you wanted to have a pet extremophile 𝘎𝘕𝘯𝘕𝘴𝘪𝘴, you would have to have that pressure cooker on 24/7 because it's going to fall apart in our world. So here is God saying, I'm going to create life as deep as you can find it, and even high in the skies also. And they're abundant. Our second point is they're abundant in all ecosystems. So here are creatures called lichens. We can find lichens growing on trees. There's a picture of a tree in Michigan, my homeland, and growing on even dead trees and live trees. And here we find them near my home in California now at 7,000 feet in the high desert, growing on a rock. What are lichens, and what do they have to do with bacteria? Lichens are associations between a fungus and an algae. And if we took the fungus and algae of the lichen and we took it to a laboratory and separated them, they would just look like mush. But when we put them together, they form plant-like structures, cups, sometimes hairy lichens, leaf-like lichens. They're shapeshifters. The algae is getting energy from sunlight, and the fungus is helping to form these shapes and colors. This is on that rock in California. We have these are lichens in brown, lichens in bright green, in gray, and the cups. This was in the dead of winter at 7,000 feet, and they survive in summers where there's hardly any water. What are they doing? They're actually breaking down this rock, mineralizing it very slowly. So the minerals are leached off this rock. And now you can see a plan here for this creature. What I didn't mention is bacteria as part of this creature. What we have found only recently is that bacteria are a third partner in the lichens, and it's a required partner. So what are they doing? They're helping with decay cycles on Earth, but they're also breaking down rocks so that the minerals can go in the soil, and those minerals are enjoyed by plants. In fact, I live near the Central Valley in California, one of the best areas to grow things in our nation, where a lot of our fruits and vegetables come from. And there are scientists who believe that these lichens have contributed over the years to the minerals that go down into the Central Valley. So there's the biomatrix. There's the biomatrix right there, with bacteria being involved, growing on rocks, growing on trees. Sometimes we don't even really, they don't even catch our eye sometimes. They're just there. Let's look at how bacteria recycle nutrients locally and on a global scale. And this is quite remarkable. This is a bacterium called cyanobacteria. You'd think it was an algae. And in fact, when it was first discovered, it was thought to be algae, but it is a bacterium. And there is so much cyanobacterium in the oceans that it contributes 50% of the world's oxygen. Okay, just as much as plants on the land. So remember, photosynthesis, what does it do? It takes in carbon dioxide and it strips the carbon off to make sugars inside the plant, and it strips the oxygen off as releases it as a waste product, so to speak, to the plant. But humans and animals use that oxygen to survive. So, but it's interesting. They can't do their job very well unless they have a helper. So again, they produce 50% of the oxygen on Earth. This is a virus. And viruses that invade bacteria are called phages. So I told you that some viruses can have these interesting spaceship-like shapes. It has a head group here where the DNA is. It has a structure right here, actually an injection tube, and it has these tail fibers. And it actually lands on the cyanobacteria with its tail fibers, and it helps to pull down that tube, and DNA is put into the virus. To do what? To basically grow new viruses that eventually explode and break open the cell. Well, how is that a helper? Well, actually, it doesn't explode every cell. It is involved in population control, but for some reason, a good number of bacteria, it does not explode. It actually injects DNA that can help that bacteria. You say, what do you mean? What do you mean? Well, think about what would happen to you if you went to the beach and you laid on the beach all day, or you took your raft on the ocean or the lake and you laid out, okay, for hours. What's going to happen? You're going to have terrific skin damage, and you might not even survive that amount of light. So what this bacterial virus, which you call a phage, all bacterial viruses are called phages. This phage can actually restore sunburn proteins in the cyanobacteria. It inserts its DNA, the DNA is interpreted and said, oh, this is a protein we need, and it reinvigorates the cyanobacteria to undergo photosynthesis. An incredible, incredible story. Let's take a look from a satellite. Here's a satellite image of cyanobacteria in Lake Erie by Michigan and Canada, and we can see the green blooms here. They're called blooms. We can find these blooms from satellite images off the Ivory Coast of Africa. They're everywhere in the ocean. And so I think we could really make a case that cyanobacteria are the most numerous bacteria on Earth, producing 50% of our oxygen. And they say the cyanophage activity is so high that it's suggested that every 7th or 8th breath we take that oxygen that you breathe in was assisted by a phage. It's kind of interesting to me that God takes this weakest creature on Earth, and he allows it to help support our lives. I think it's just like him, his testimony, that the weak can be strong. So, what have we talked about? We've talked about bacteria abundant on Earth and all ecosystems. Bacteria recycle nutrients locally and on a global scale now. This is part of that biomatrix idea that we're looking at. But they could also act as symbionts for humans and many animals and plants. Let's take a look at this. Let's look primarily at humans. First of all the 35 trillion bacteria that we have live on our surfaces. So what do you mean by that? Well, there are two surfaces that we present to the outside world, our skin and our gut. Think about it for a minute. Your body is kind of like a hollow tube. You have your skin on the outside, and on the inside you have your gut tube, and that really is on the outside of you also. So those two places are where most of those 35 trillion bacteria reside. You're pretty sterile inside. In fact, you don't want some of those microbes going inside because your immune system might get confused about what's going on. So in fact, in the intestine, the bacteria are called the microflora. So this is kind of another word that is a companion, similar to microbiome. It's just another word that it's often used in medicine. They talk about our normal flora. That's our normal microbiome, or microflora. So we have 10 to the 14 microorganisms in the human intestinal microflora, and they live on the top of on the outside of your gut, okay, in your stomach, but on the outside layers. They typically don't get through because of an amazing layer called the mucus layer. Mucus is something we don't think of very positively a lot, okay? But it is an amazing protein that actually keeps those bacteria away from getting inside the body. Even more so, your immune system can reach up across that. You have white blood cells in your gut that'll reach up across that mucus layer into the layer of bacteria and say, you're in the right place, you're in the right place, I'm not going to send out an alarm. And they calm down. That's going on every minute, minute by minute. So you have this intestinal microflora or microbiome in your gut and on your skin. Let's look at your skin for a second. We have bacteria that have acne in the name. And we know that acne causes skin blemishes on our skin. Sometimes when we were teenagers, we struggled with this. And most strains are helpful. They're probably outcompeting pathogens. But there's one called Cutibacterium acnes subspecies that can cause those blemishes. So a few years ago, they found a chemical to treat this called isotretinoin. And they found out that not only does it destroy the bad Cutibacterium acnes subspecies, but it lets the good species outcompete them. That's interesting. That makes sense. The biomatrix not being totally destroyed. It's working as a barrier. Then in fecal transplant, these have worked clinically. What do we mean by this? You can get some bacteria in your gut that can be invasive. They can get through that mucus layer, and then when the immune system sees them, it says, kill. And now your immune system's overreacting, you have an infection going on, you have damage. This ultimately can be lethal. So what happens is we can take fecal tissue from a donor and put this back in the human body and outcompete and displace those wicked bacteria. And this has saved lives. This has saved lives. Doesn't sound fun or a pleasant thing to think about, but it actually works. Also, we talked about the mucosal barrier and the immune system interaction. We already talked about that. How that's amazing that the immune system, it keeps track on where those bacteria are. So the mucosal barrier doesn't work alone. Let's look at something related to this called the gut-brain axis. This is fascinating, and it's fairly new. We now know that the bacteria that live in our gut, there's different species, E. coli is one of them, they make factors that support our health. And some of those factors can make it to our brain and influence us for good or for bad. So in your gut, a lot of us have bifidobacteria, and they produce tryptophan. And you've heard of the amino acid tryptophan. We often get it from eating turkey, and it kind of makes us tired and willing to take a rest, which could be a good thing. But we have bacteria that produce this, and this gets to the brain. We have gut microbes that produce short-chain fatty acids. These are messenger molecules that have effect. We're still trying to figure out what they do, but they make it to the brain. I'm going to skip this third point and come back to it. I'm going to go down to here. Transferring gut bacteria from humans to mice is something we've tried. So to study this gut-brain axis, we have taken bacteria from people with milk allergy. We put them in the mouse gut and they get milk allergy. That seems logical, but the next two seem more fascinating. We can take bacteria from a long-distance runner, put it in the mouse gut, and that mouse will run on its treadmill farther and longer when it has those human gut bacteria. We have taken gut bacteria from people who are depressed and put it in mice, and they've done tests of mice, and they're sleeping a lot. They're actually acting depressed. So something's going on here with this gut-brain axis. It's kind of in the middle of its research tenure, and hopefully some good things will come out of it. But let's go back to this third point right here. There are some E. coli strains. Remember, E. coli is a bacterium. We find it in our gut. We hear about it a lot. Not necessarily the most dominant gut bacterium, but one of the most studied because we use it in research. There are E. coli strains that can produce tangled fibers, and they can make it to the brain. Now, what's important about that? Well, if we look at Parkinson's disease and Alzheimer's disease, they are both killers of the elderly in our society. I had a parent suffer from this. It was hard to go through with Alzheimer's, and it took that parent's life. And thankfully, they lived a long life, but it's hard to watch. What we know now, there's a mouse model, and this is an incredible podcast I watched about a researcher. He put these E. coli strains in mice that made tangled fibers. So let's back up for a minute. What are tangled fibers? If we look at Alzheimer's and some other brain conditions, what we find is the degradation that occurs is between neurons. So you have nerve cells that actually talk to each other by binding to each other. And they have long structures of a single cell, and they end in these dendrites that interact. In these dendritic areas and these long areas, we have buildups of tangled fibers in Alzheimer's. It's striking. If you look at the pictures, it's just buildup of protein fibers. And as probably you can guess, it's breaking down communication and it's causing illness. We still haven't figured it out. There's other factors that are going on. But this researcher took, in a mouse model, he found E. coli that produced tangled fibers. He put it in the mouse's gut, and he had the mice walk across a little bar in their nest. And those who had the tangled fiber E. coli would fall off more frequently. So now he has a model that suggests that these tangled fibers do cause this problem, and now we can study that to possibly look for cures. And this is fascinating. So this is, again, called the gut-brain axis, and I think we're going to find out more about this. You know, maybe your mother said you are what you eat, and maybe there's a lot of truth to that now as we think about this gut-brain axis. So why are there bad microbes in God's perfect creation? Let's look at some ideas here. We know that God filled the world with life, with living microbes supporting life on Earth. And we see how he did this exquisitely with microbes. We see that testimony before us when we see microbes in the biomatrix. Bacteria show exquisite design and have a purpose in nature. This certainly suggests that they were created for a purpose, and now we're finding some of those purposes as they assist in human health and in producing things that we can take advantage of on Earth. Bad bacteria are probably they have to be the result of the fall. With our theology, there's little other ways to think about it. So they probably are the result of the fall. But I want to end with this. Even in pandemics, God's goodness and plan can be seen because there are survivors. God does not want humanity to be wiped out before he returns. I had a friend say, there's a person that said COVID is going to kill humanity. Well, is Jesus here? Is God here? Are we in the end times? And so this is interesting to think about. But I want to go back to that book on pandemics, because the author says something incredible. It was not meant to be a Christian textbook. Here's what he says. He talks about the Roman plagues of Cyprian and Antony, and he believes, the secular writer believes, that they probably helped to spread the gospel. Here's why. How do we, here's a quote from the book. How do we explain the sudden transformation of Christianity from a marginal Jewish sect to a popular religion? American sociologist Rodney Stark argues that infectious diseases are a crucial part of the story. Another quote, the Christian faith skyrocketed because it provided a more appealing and assuring guide to life and death than paganism. Fascinating remarks in this book. Christians would have been able to reduce mortality by 2/3 just with basic nursing. This is a testimony to the Christians during these plagues who are risking their lives to take care of their neighbors. In Roman culture, it was basically every man for themselves. Last quote, the fact that so many Christians survived and that Christians managed to save pagans abandoned by their families would have provided the best recruitment material any religion could ask for. So here we have these plagues where hundreds of thousands of Romans and Greeks and Jews are dying, and the Christianity starts to surface as a movement because they are people willing to help those who are dying. So this is the end of our lecture, and I appreciate your attention.